Have you ever pondered the intricacies of how cells duplicate their DNA? This process, known as DNA replication, is a fundamental biological mechanism that allows for the faithful transmission of genetic information from one generation of cells to the next during cell division. It's a marvel of nature's engineering, and it occurs during the S phase of the cell cycle in eukaryotic cells. The process begins with initiation. DNA replication starts at specific sites on the DNA molecule. These are known as origins of replication. In eukaryotic cells, there are multiple origins along the length of each chromosome. Enzymes called DNA helicases unwind and separate the double-stranded DNA at the origin, forming what's known as a replication bubble. Next, DNA helicases further unwind the DNA double helix, creating two single-stranded DNA templates. Single-strand binding proteins, or SSBs, bind to these exposed single strands, preventing them from reannealing. Following this, an enzyme called primase synthesizes short RNA primers complementary to the single-stranded DNA. These primers provide a starting point for DNA synthesis by DNA polymerase. Now, we move on to the DNA polymerase binding. DNA polymerases bind to the RNA primers, marking the beginning of DNA synthesis. DNA polymerase synthesizes new DNA strands in the 5-to-3 direction. During DNA synthesis, the leading strand is synthesized continuously in the 5-to-3 direction as the replication fork opens. The lagging strand, on the other hand, is synthesized discontinuously as short fragments, known as Okazaki fragments, in the 5-to-3 direction away from the replication fork. DNA ligase then joins the Okazaki fragments on the lagging strand, creating a continuous complementary strand. But what about mistakes? Well, DNA polymerases have proofreading capabilities that allow them to correct errors during DNA synthesis. Mismatch repair enzymes scan the newly synthesized DNA for errors and replace incorrect nucleotides. The process of DNA replication continues bidirectionally until the entire DNA molecule is replicated. Termination sites exist, and when the replication forks meet, the process is complete. In eukaryotic cells, there are special structures called telomeres at the ends of linear chromosomes. The enzyme telomerase helps replicate the telomeres, preventing the loss of genetic material during replication. Finally, the replicated DNA, now consisting of two identical sister chromatids, is packaged back into chromatin. The cell is then ready for the next stages of the cell cycle, including cell division. So that's DNA replication, a highly accurate, coordinated process that ensures the faithful transmission of genetic information. The accuracy is maintained through the action of various enzymes involved in proofreading and repair mechanisms. The resulting two daughter DNA molecules are identical to the original DNA molecule, and each daughter cell receives one copy during cell division. A true marvel of biological engineering.